Today's speakers are Laura Hamuru and Joga Gaburu. Laura is a senior research investigator at Bristol Myers Squibb in clinical pharmacology and pharmacometrics. She earned a PhD in biochemistry and molecular biology from the Pennsylvania State University College of Medicine. Her master's degree is in pharmacometrics from the University of Maryland. Joga Gaburu is a professor in the Department of Pharmacy Practice and Science and director of the Center for Translational Medicine at the University of Maryland. He earned a bachelor's of pharmacy and a master's in chemistry from the Berla Institute of Technology and Science. He has a PhD in pharmaceutical sciences from the North Dakota State University. And he has an MBA from Johns Hopkins University. Laura and Joga, welcome to today's event. I'll now turn it over to Joga to begin the presentation.
Well, thank you all for dialing into this timely and relevant topic of drug development against rare diseases. The crux of the matter that you will hear during this webinar is essentially a choice between empirical approaches for establishing substantial evidence of effectiveness versus I would categorize as more efficient approaches. Some people may call it model-based, but that turns people off. So let's focus on the virtues. It's more efficient. That's what we will be talking about today. And if you have not reviewed or had not had an opportunity to review this publication, I request that you consider it. It's a very easy read, couple of pages, and it kind of goes into details about what I'm going to say in the next couple of minutes. And the challenge is, if for a non-rare disease drug development, the resources in terms of access to patients is enormous compared to drugs against rare diseases, which means I can take a lot more chances to get my trial right, and I can repeat that until I can get a drug meet the efficacy standards. Now, if I have to do the same type of approach to developing drugs against rare diseases, unfortunately, I cannot do it because there are not enough number of patients to start with. So that means in the equation of power to show efficacy, I have three elements. One is the effect size, the second is the variability, and the third is the sample size. Now, where most of the drugs for non-rare diseases, you would see, their effect sizes are not stellar. They're, modest. But my sample size, I have freedom. I can study, large number of patients. That is out of the equation for rare diseases. So I'm stuck with then two things. One is effect size and variability. Variability, maybe we can understand better to screen patients and such and come up with enrichment designs. But still, you're starting with a very small sample size. So enrichment, I don't know if it's practical. Then you're stuck with effect size. So that means inadvertently, the current standards for approval of drugs against rare diseases the standards are much higher for rare diseases, I argue, than non-rare diseases. Because for me to show a drug is efficacious, I really have to have a very big effect size given my small sample size that I can recruit into the trial, which has led to controversies in terms of the recent approvals in the United States. And there are people who agree with that approach of the so-called courts lowering the standards, as some deem, versus some other folks think that's a prudent or a pragmatic approach to supporting drug development and access of such drugs to patients, albeit only some of them could benefit from these drugs. So that's a debate. I'm sure you're all familiar with it. And here we are talking about, in this webinar, on the more efficient approaches, meaning how do I look at the data or knowledge available for a given condition and use that, leverage that knowledge to make the drug development more efficient. Can I use longitudinal data instead of using end of the trial changes? Can I use can I assume some things, meaning the shape of the model? By assuming the shape of the disease progression in this case and or the drug effect, I can get some additional power in my analysis, thereby making the drug development more pragmatic and feasible. So that is what you would hear from Laura, and then we can engage back in a discussion. So I turn it back to Laura.
Thank you, Joga, for that really important introduction. It's really making us think that we have to approach drug development for rare diseases and our clinical trial designs in a very different way. And I think you really illustrated some good examples of that. Before I get started, what I wanted to do is thank Suzanne Mitten from Certara. She really helped to evolve this webinar, and her support for broader communication of disease progression modeling for rare diseases is very appreciated. And I want to thank those who dialed in today, the audience, for sharing your lunch time with us. Some of you may be in the rare disease space. Some of you may be developing disease progression models. So thank you for attending, and I look forward to a Q&A after the presentation. So if we go on to the next slide. So during this webinar, I'll give a little bit of background on Duchenne muscular dystrophy and go over some of the motor function endpoints that have been used in prior DMD trials. And I'll discuss, because this is a pediatric disease, there are childhood development contributions to these motor function endpoints, and I'll go over that in some detail. And then also kind of highlight a structural approach for how we look at the data, what decisions we hope to get out of this disease progression model to really inform downstream trial design and looking at drug effects. I'll summarize by going through some predictions from the disease progression that we've quantified, and then use the model to simulate a potential drug effect for a future trial. So Duchenne muscular dystrophy is a life-threatening disease. It's a rare disease that affects boys as it's sex-linked, and Duchenne is unique among muscular dystrophies in that there's a loss of function mutation in the dystrophin gene. And it turns out the dystrophin protein is pretty important for the integrity of the muscle, forming the network with the actin-myosin skeleton with that of the subcutaneous space. In addition, it's thought that the dystrophin protein coordinates signaling complexes that really improve signaling and growth and differentiation of the muscle tissue. This is a progressive muscle weakness disease and eventually functional loss. Children are usually diagnosed about 4 to 5 years old after they fail to meet certain developmental milestones. And unfortunately, by the age of 12 to 14, the patient loses the ability to walk. In the mid-20s, the disease really takes over, and the patient succumbs disease. So it's very life-threatening. There's corticosteroids as the current standard of care, prednisone and dafloquart, but there's really a need to address the key underlying issues of the muscle atrophy and defects. And to that end, over the past several years, there have been several approaches reported and some approved now as drugs for DMD patients that focus on replacing the ineffective gene, therefore making the dystrophin protein. Some of these therapies, exon skipping therapies, stop codon read-through therapies, and also gene therapies to actively replace the gene for dystrophin or fragments of the gene for dystrophin. So it's a promising time in the field of therapies for Duchenne's. The clinical outcome measures that are used to assess drug effect in trials, this is a bulleted list of some of those that have been used historically. The first two are distance measures, and the last two there are time function tests. There are also composite scores, such as the North Star Ambulatory Assessment, and this is comprised of 17 different evaluations of upper and lower motor function. What I want to focus on is the 6-minute walk test, but what I'll be describing, in theory, could be applicable to these other motor function tests. So it's been recognized for many years now, because this is a pediatric disease, there's a developmental component to how children perform in various motor function tests. And what this slide is illustrating on the left is the 6-minute walk test versus age of the patient. And on the right is the composite score I described versus age. And one striking feature you can see, subjects in the lower age range actually are showing some improvement in motor function, both in the 6-minute walk and in the composite score motor function test. However, there's this point where the disease takes over and there's this more precipitous decline in how they perform in the motor function. Now, this dependence on age is important when you're designing a trial to assess drug effect. For example, if age isn't considered in the trial design, you can imagine a situation where subjects at lower age are enrolled in the placebo group, and they're going to perform relatively well and show improvement. However, if you primarily recruit older age children into your drug treatment effect, they're predicted to perform less well. So you may actually miss a drug effect simply due to your age bias. Conversely, the flip side of that would be if you recruit younger children in your drug treatment arm and older children in the placebo, you may falsely conclude that there is a drug effect. And this is very important, and it's been recognized for a while, this source of developmental age variability. So if we take a closer look at the variability component here, as bucketed by the 7 years old, and historically it was felt from observational studies that around 7 years old, children start to show the decline in performance. The top lines here show the 6-minute walk test performance from the McDonald et al. Manuscript. And what I want to point out is you can see the trend for the mean values at 6-week intervals up to approximately a year are showing improvement in performance. And the variability around this performance is relatively constant. So this is a 1-year trial, so it's roughly 7 to 8-year-olds. However, at the bottom panel, subjects that are greater than 7 in this observational study, you see this mean trend for reduction in 6-minute walk test performance, and you also see really high variability both at baseline and then at the 48-week interval. So there's this discrepancy in variability across the age buckets here. So to further illustrate this variability component and the challenge with demonstrating a drug effect, These are plots taken from an FDA briefing document, an AdCom meeting, April, almost 2 years ago now. And on the left plot, what you see is the 6-minute walk test versus the trial duration. The red lines are the therapeutic treatment arms, and the black are the natural history control data to evaluate the drug effect. And on the left, what your eyes are looking at is a trend for improvement with the drug treatment. However, when you plot the data versus the age, that difference becomes less obvious. And the question is there a drug effect? And this is certainly one aspect of how you evaluate drug efficacy, and an important one because it's assessing motor function and endurance. But is there a way we can use more quantitative approaches to discern a drug effect given this age-dependent variability? So what I'm highlighting here is when we initiate a kind of a quantitative approach to a problem, what is the decision we hope to get out of the work? It's an extensive amount of work, and these decisions can fall into buckets of go, no-go with the drug at certain milestones. These decisions could impact how the trial is being designed, how we're going to inform the label, and what are the key questions we hope to gauge from our quantitative approaches. And in this case, these were really decisions around how to design the trial and evaluate a drug effect. The information we had, as Joga had mentioned, patients are very limited and precious, and getting access to data is a challenge. We can look to the literature, and fortunately there were several really nice manuscripts from observational studies describing the natural course of several of these motor function endpoints with time. And what I was specifically looking for is literature data that characterize longitudinal information for individual subjects as they age. And this was really to try to capture the intersubject variability in the model. And then the analysis methods used are to use mixed effect population approaches, again, to bucket the various forms of variability, and then have a quantitative framework that we can then use to simulate different drug trial scenarios. So where did we get the data? This is a summary table of the data used in the analysis. This work was published last year in CPT-PSP, the majority of the work that will be described, so you could access more details around this analysis. What this is showing in the first 2 rows are the manuscripts where data was digitized to build the disease progression model for 6-minute walk test. And the 3rd row is a manuscript. Data was digitized but not used to build the model, but used to evaluate the accuracy of the model. And what's listed in this table is just source information from each of the trials. So McDonald et al. Was a placebo-controlled trial. Gomans et al. Was a natural history study from the Leuven site in Belgium, a very homogeneous population. I'm indicating the steroid use here. What I didn't describe is the steroid use can actually delay the loss of ambulation. So having that information, even though it's at a kind of an aggregate level, was important to understand. In fact, I looked specifically for data where the majority of the subjects, if not all of the subjects, were on steroid, given that that was the standard of care, or is the standard of care. And this is just further information here in the columns on the mean age and some metrics around the data itself. So using, actually what I wanted to emphasize here, because it's important to do, literature data is very valuable, but along with that comes some limitations, and this is just a reminder of those. We pulled data across 2 different manuscripts to kind of get at this first bullet too, the accuracy of reporting. But that's something to keep in mind when you are using data from the literature. The patient demographics information, you don't have. You have aggregate level information on age, race, weight. You have longitudinal on the actual 6-minute walk test, but not detailed information on the patient demographics, which really limits your ability to build the disease progression model. And thirdly, when you use data and you don't have the corresponding information on the patient demographics, co-medications, detailed information on the type of steroid, or other patient factors that you could imagine would impact disease progression, such as the dystrophin mutation, baseline cardiac and respiratory function. Again, it limits your ability to really parse out the forms of variability or the components that are really driving the variability. But it's better than no data, and it's a platform that you can grow and build with your program. So this is a plot showing the digitized data from the McDonald and the Gomans paper. It covers 86 subjects and includes 228 observations. In general, each patient is contributing 2 observations. Several subjects, over half of the subjects, have greater than 2 observations captured in the data set. So the modeling strategy here is to come up with a structural model or a way to describe the up and down nature of the data. Let me just go back a second to really emphasize that in the data here. At the low age range, we have subjects that are in an upward trajectory, some not. It's a very heterogeneous population. This subject, you can kind of even see their maximum, going through the maximum of decline, where they peak and then start to lose 6-minute walk test distance. So in order to apply a quantitative approach to this, we had to look at a series of different models to kind of characterize this up and down nature. This wouldn't fit to a standard linearization. So to do that, we kind of walked through different structural models, and I'll go through the model that we ended up selecting. Once we had the model in place, we wanted to evaluate the model using some of our common tools, visual prediction checking, but also simulate different scenarios and compare the model predictions with that produced in the literature. And then finally, we can apply the model by simulating a potential drug effect. So again, our criteria was really to come up with a quantitative way to describe the curvature in the data, going through the maximum. And we can use our statistical outputs to evaluate model improvement, and then look at some other criteria of the model to assess whether or not the model adequately characterizes the data. And then finally, interpret the parameter estimates that are coming out of the model. Are they meaningful? Are they interpretable? This is a list of the models that we kind of walk through, listed in increasing complexity, starting out with very linearized approaches, but then putting categorical effects such as age on the slopes and intercepts. And for the first 2 models, there was a requirement in order to capture the inflection to fix the age. There was historical information suggesting this was around 7, but what we wanted was a model where we didn't have to a priori fix that age. We wanted to have the model speak for itself and let the data speak for itself to estimate what that would be. The quadratic equation is a structural model that could capture that. However, this structural form is not very stable. We looked to our colleagues at Certara, and Certara provided a lot of help in kind of thinking through alternative ways to develop quantitative approaches to characterize this maximum. And we landed on a model which is really a series of linear models, but rather than fixing and dictating which data points go with which linear model, we could have the model decide that, and I'll go through that in the next slide. that was the model that we ended up selecting for downstream work. Two other approaches, the Bateman function, gave a reasonable estimation. The parameters from this model are hard to interpret the meaning. And then finally, a more complicated model, fitting it to an indirect response model. On the next slide, I'll go through the Phoenix NLME code for the linear simultaneous estimation. And what this is showing here is you can describe two linear equations, and then you can have the model minimize the response for the first linearized function and then the second linearized function. So the model can effectively minimize the up and down response trajectories. We can include inter-subject variability estimates on the slope. This allows us then, as a secondary parameter, to calculate the age at which both the population and the individual trajectories begin to decline. And then finally, having this framework allows for assessing additional forms of variability in the model, such as covariate effects on the slope or intercept. So these two plots are illustrating, on the left, the population fit in blue of the model and the observed data, and on the right are the individual fits as a function of age. And the age at maximum for the population fit is roughly 10 years old. And that was an interesting finding out of this model because prior work had suggested a much lower age. You'll notice on the individual fits on the right, there's various degrees of inflection. Each patient starts to decline over a range of ages. And this will be illustrated in the next plot, which is the histogram across all subjects, with the mean age of decline listed with the red bar. So there's a distribution of ages where subjects begin to decline, with a mean age of roughly 10 years. As a reminder, in this data set, roughly 80% of the subjects have been on stable steroid use. And that is an important piece of information. This shift in 10 years old, because there was a disconnect with historical data, and we were using a more unique approach, let's say, to do this model estimation, I wanted to look back at the literature to see if this actually made sense. And there was a paper in 2015 that looked at steroid use and how it impacted the loss of ambulation. And what I noted here in one of the figures, subjects that weren't on steroids or intermittent steroid is the red. And subjects that were on stable steroid regimens for greater than 1 year is in the black. And there's approximately 3-year delay in the loss of ambulation. And that number was significant because what our model was saying is there was a shift of roughly 3 years and when subjects started to decline. So there was some further support with this external data in what the model was predicting. So to further build on that, I looked through some older literature where maybe steroids wasn't the standard of care and digitized that data and then used the model to fit that data. And what I found, the model is actually predicting a lower age range, more consistent with the historical data. So this was a further validation that it wasn't the model biasing in some way, this higher age prediction at which you would start to decline. But perhaps in the earlier data sets, this in fact was true, that 7 years old was the mean age of decline, and that the use of steroids more widely implemented was in fact delaying this. And the data used as a reminder to really build this model, over 80% of the subjects were on steroid. So there's self-consistency then in what the model was predicting. So if we look at the details, and one of the advantages in using a linearized approach is there's a more direct interpretation of the parameter estimates in the model. And this is just delineating here the slopes and intercepts, the first developmental slope, and then the disease-induced, what I'm calling the disease-induced slope. The model is predicting that in the development phase, subjects will perform, show an improvement in performance in the 6-minute walk by 20 meters per year. This is in contrast to healthy boys over this age range, which actually show about a 40 meter per year, not shown here, but detailed in the manuscript. The disease-induced slope, the model is predicting that once the patient starts to decline, they'll see an 85 meter per year loss in performance in the test. And the error contribution in the model is roughly 57 meters. It's a high variability component. This is an additive error model. But recall, this is including all age subjects, and it can run as high as a standard deviation of 90 up to 150. So the model has done a pretty good job of accounting for some of the age-related variability. And at this point, in this form of the model, that's all we're capturing is the age-related variability. So our conclusions here was doing a fairly good job of accounting for the majority of the variability. So if we go to the next slide, and once we had the model, this is something that's commonly done, is to repeat using simulations the variability components in the model with the parameters fixed and ask the question, can the model reproduce the original variability in the data? And what this is showing is after 2,000 replicates, the observed quantiles in red and the predicted quantiles from the model in black. And what I want to point out, up until around the age of 12, the model does a fairly good job of predicting the observed data. But after 12, there is this deviation, and the model is predicting a more precipitous drop. So that really highlighted to us that up to 12 years old, we can potentially have reliable information coming out of this model, but greater than 12, probably not. We really need to have more data in the higher age brackets to develop more confidence in the model. But that's okay. We can confine our assessment to 12 years and younger. So again, going back to the original data sets in the first 2 rows that we used to develop the model, and then the third data set to really kind of an independent verification on the model predictability. So when we look at the 2 data sets that we used to build the model, on the left is showing 6-minute walk test performance, and this is represented as a change from baseline as a function of the trial duration. And in red is the endpoint from that study at the 48-week time point, and the black is the model predicted. And the model is doing a fairly good job of predicting back the 48-week endpoint. In the Goldman study, again, we're showing model prediction versus the observed data reported in the manuscript. And a fairly good job, the model is doing a fairly good job of predicting the observed data. This perhaps is a low-hanging fruit, right? This data was used to build the model. What about taking a third data set? And to that, we looked at a paper by Mazzone et al. In 2011. And what I'm illustrating here in the first row is the Mazzone data predicted just using the age demographics, the aggregate age demographics with the min and max range from the manuscript, and running it through the model and asking, what would the model predict? What would be the change from baseline 6-minute walk test performance across all ages, and then I did the historical stratification because the paper actually had a breakdown of the less than 7 and greater than 7-year-old age groups. And across all ages, the model is predicting fairly well the observed data. If you look at the age breakdown, model is actually over-predicting the performance in both cases. In the less than 7-year-old, predicting an improvement, whereas the observed data is showing a loss. In the greater than 7-year-old, less of a loss than the actual observed data. And trying to reconcile, what does this mean? And looking at details of the Mazzone paper, only half of the subjects were described as being on steroids. And this points to limitations of literature data, not knowing, too, which subjects were on the steroid, one, and not having enough detailed information to adequately compare with the observed data. One hypothesis as to why the model is over-predicting performance is that the model was built on subjects that were on steroid and were presumably improved in their abilities to perform on this test, whereas the observed data, they weren't. In all, we still had confidence that the model was doing a fairly good job of predicting performance on this test. And as you're building the models and validating them, the idea is they grow with your program. So you're always coming back and updating with data, and you're assessing the model and other variability components in the model. So with the model we had developed from the literature data, we wanted to ask the question, can we observe a drug effect? Well, we didn't have any data where we treated subjects and had the corresponding 6-minute walk test. But what I wanted to show, actually before I went into the model description, if we were designing a trial, and this gets to Joga's comments at the beginning, and we have a drug effect, and in the McDonald paper they reference 30 meters as having clinical meaningful effects. So if our drug could improve performance by 30 meters, that was deemed as a meaningful result. If you would do a standard sample size calculation to detect a drug effect, in green is less than 7 years old with a 40-meter standard deviation. You would run a trial with roughly 40 subjects with a 5% type 1 error rate. If you looked at an aggregate age group, and the goal in rare diseases too is to treat as many people as possible across the age range, you would have to recruit 160 subjects per arm in order to see a drug effect. And these are very high numbers for this rare disease. This illustrates that we need alternatives in the rare disease space. So what if we use the model and leverage the disease progression model where we have age explicitly characterized and the age dependence to 6-minute walk test variability. And we conduct a trial where we randomize the ages to different placebo versus treatment panels, and we allow all subjects across the broad age range. And we mimic a drug effect. Placebo would have the initial parameter estimates in the model, and the low and a mid and high would have an incremental 30-meter improvement in development, as well as a 30-meter reduction in decline. And then ask the question, can the model actually detect this drug effect? And so we use the model to simulate 100 trial simulations at the 6 subjects per treatment arm. And then we looked at the data to see if a drug effect could be detected. And what this is showing is a predicted dose response for this hypothetical drug. Again, change from baseline in the 6-minute walk test performance over to 1-year trial duration, and then looking at the placebo group and then comparing that to the low-dose treatment arm and running statistics indicating that we could detect a significant difference if we use the model to parse out the age dependence of the variability. So I wanted to summarize. We were able to develop a 6-minute walk test disease progression model that predicted the patient age on average would experience a 20-meter per year improvement during this childhood developmental phase. The model also predicts that during the more precipitous decline, subjects would lose about 85-meter per year performance. The mean age of decline is predicted to be 10 years old, which is approximately 3 years or 3 years older than the historical age. And this is hypothesized to be due to the increase in steroid use for some of the later observational studies. The trial simulations predict that the model could detect a 30-meter treatment effect, even in a small n-number trial that was not a priori age randomized. And finally, what we've done is set up a platform, a quantitative platform that we can use to build in other sources of patient variability that would likely drive additional patient variability, such as the dystrophin genotype, lung function in the subjects. And in theory, these other sources of patient variability could be included in the model and evaluated in terms of their significant contribution to the variability. So what we've done is establish a platform that can be used for ongoing work. And as Joga was describing in the introduction, you really don't get second chances. Patient numbers are low, and you really want to do a good upfront job of understanding the endpoints that you're using in the trial, what's driving the variability, and designing the trial in such a way you maximize your chances of detecting a drug effect. I do want to acknowledge, importantly, the DMD patients. Data was reported in several publications that I used. And I want to thank the authors of these papers. Without this work being published, we certainly couldn't have built on that data, and that was very important. I want to thank past and present Bristol-Myers Squibb employees. Malaz Abu Tarif has been a really good mentor and friend, helping to establish early parts of the model. Phyllis Chen was very integral in coming up with the idea to really look at literature for this particular disease to develop a model. And Gary Tiracere, who was leading this program. Also like to thank Greg Thalhauser and Helen Moore for very useful discussions on model development. I'd like to thank University of Maryland. Joga has been a strong teacher, mentor, and inspiration. He's done a lot of work, as the audience knows, in pediatric world, in rare diseases, and really has been an inspiration. Masangi Gopalakrishna really was integral in teaching me a lot about statistical understanding and sources of variability in her statistics courses at the University of Maryland. And Vijay Ivaturi, who was phenomenal as a teacher and taught me a lot of very interesting techniques in the pharmacometric space. And finally, Certara. Christopher Mell was very helpful there was a lot of email communications back and forth, and I really appreciated the very rapid turnaround with the Q&A from Christopher and the Certara group. And then finally, Suzanne again for reaching out and developing this very informative webinar. Okay, it looks like it's 11:45, so we have plenty of time for Q&A. If you have questions, please submit those now.
Wonderful. Thank you so much, Laura, for that very informative presentation. We do encourage our audience, as Laura mentioned, to submit their questions to her in the Q&A box. Laura, it looks like we have a first question from our audience. Someone wanted to know, sorry, just bear with me for a second. Are there any age-dependent data on how muscle size is lost and muscle composition changes in the Duchenne muscular dystrophy population during the course of disease progression?
Yeah, so there's known atrophy happening in the muscle itself. I'm sure there are imaging techniques to look at actual muscle versus fat, and potentially using some of that data on muscle physiology to further understand the age dependence. I'm not aware firsthand, but it's likely there is information on the actual muscle physiology or in this case muscle size and the compositional changes, as you indicated. That would be interesting actually to look at. You know, as another potential source. You can imagine a larger subject with bigger muscle mass may have a different trajectory than someone with a lower muscle mass.
Someone wants to know, can this disease progression model be used for other developmental endpoints in Duchenne or other pediatric diseases?
Yes, in theory, when you have this up and down change in a muscle function, motor function test, you could apply this same model and re-estimate. And, you know, even other developmental increases and decreases, data sets that you have, you might want to consider using this linear with simultaneous estimation. That definitely could be used.
Someone else wanted to know, Duchenne muscular dystrophy is typically thought of as a progressive disorder. Can you comment on the model prediction and the improvement in the 6-minute walk test? Is that due to the younger age of the patients?
Yeah, so that developmental slope that we have defined in our model is predicting a 20-meter improvement up until the mean age of 10. So 10 and lower on average, the model is predicting they would improve at that rate. I mentioned briefly that healthy boys, McDonald has done a nice study, had a nice manuscript describing Duchenne and Becker patients relative to healthy boys over this age range. And healthy boys actually perform on this test roughly double this, so 40 meter per year, up until about 8 or 9 years old. And then there's a plateauing effect as the muscles develop. But in Duchenne's, there's this initiation of this more precipitous decline. This model is predicting around 10. These patients have been on steroid regimen. Earlier work suggesting a lower age where this precipitous drop happens at around 7.
How can other patient sources of variability be captured in the disease progression model?
Yeah, no, that's a really good question. And immediately one of the things that could be evaluated is on the slopes. what are other factors driving this 20 meter per year versus the 85 meter per year loss in the decline phase? Is it due to, the other question was looking at the muscle size, body mass index, fat to muscle ratio, other things as a genotype, the genotype leading to the loss of function mutation, some cardiac indicators. Do patients that have more inflammatory signals have a faster descent in the decline? This model now is kind of primed to start interrogating some of these other sources, but highly dependent on having the data at hand to really ask these ongoing questions.
Looks like we have one final question from the audience. Could a historical disease progression model be used in lieu of a placebo group to demonstrate a drug effect?
Yeah, so that's another important question. And in theory, yes, there are challenges associated with that, as you can imagine. But oftentimes in the rare disease space, historical controls are leveraged. Again, it's the heterogeneity and the variables that you don't quite understand from that historical study that has the potential to confound your interpretation, albeit, but sometimes that's all you have. So the short answer is yes, with provided caveats. And I'll maybe hand it over to Joga to give us some more context to that question.
Thank you. That's a very interesting question, and that question has come up in several different areas, not just in this particular case. You can make a case that your endpoint is on an average. So yeah, there would be variability, but still on an average, does this new drug offer benefit over placebo? You could use historical controls, and that's allowed by the Code of Federal Regulations. And the question then becomes, does DMD raise to the occasion or to the level where you can rely on historical data? That means as a community, we have to do a couple of things for us to use historical data, historical controls. One is to show consistency of these, quotes, placebo effects or effects. I mean, it's not really placebo effect. It is effect, it's natural progression, I would think. It's not kind of placebo effect. So let's be clear about that. Now, and then once you see that they are consistent, we can come up with some kind of a statistical criteria to account for uncertainty. That's it's a reasonable approach. Whether the frequentists like it, we'll have to engage in a negotiation. So to me, the question is it worth it? Meaning this particular situation, DMD, I would say yes, it's a worthy cause for us to attempt to negotiate. But then there would be, if I have to argue against it, I would say there are companies who have done trials including these controls. Then what do we do with them? You know, well, they showed us that it is feasible to include placebo in their trials. We can't ignore that. That's going to work against this hypothesis. But if there is a way we can use those data also from the other trials, either through patient networks, demanding to use their data for this kind of a more, noble cause. You could use that to advantage, showing it is consistent. And there are other designs also we can consider. If there is a way that you could take patients, put them all on this drug and think about withdrawal instead of using placebo, what happens if you withdrew the drug in, shall we say, a third or 50% of the patients? Do these symptoms worsen upon that? That's another possibility. If using placebo is not, it poses practical challenges to recruit patients into the trial and such. So that's a long answer, I guess.
No, that's good. Yeah.
Looks like we got a few more questions that came in from the audience. Laura, you mentioned in one of your slides that 160 patients were needed to detect a drug effect in older DMD patients. Can you comment on what method you used to calculate it? Was it a traditional statistics method or model-based?
Yeah, so that was traditional statistical methods. Given the variability in the response, the anticipated magnitude of the effect, which was 30 meters, and the alpha of 5%. So traditional methods to get it that high number, yeah.
This looks to be the last question. Can you comment on the challenges of performing this kind of disease progression modeling?
Yeah, outside of the data, getting access to the data and having a data to work with, that was probably the biggest challenge for this project, was characterizing the data and the different forms of the structural model, but not insurmountable. Colleagues internally, colleagues at Certara, really helping to think through how to approach this quantitatively. I feel like this is just the beginning in terms of this model building exercise, that assessing covariate impacts, using this model or a form of this model to evaluate a drug effect from actual trial data. I think there's potential to do that, and that's really going to open up, in my opinion, that's where more of the challenges are going to come in, but also more of the interesting aspects and predictions from this model will come in.
Thanks so much.